Hi everyone, this is the second part of Chapter 3, Biological Macromolecules. So here we go. So just pull this up from the last presentation just to remind you macromolecules are big molecules. They're polymers of monomer subunits. And remember that we put those subunits together with the process called dehydration synthesis. We hook them together, that's the synthesizing part, by removing water. That's the dehydration part. So carbohydrates, these are your macromolecules important to organic systems. Carbohydrates, proteins, lipids, and nucleic acids. We went over those in the first part, and we'll revisit each of those in part 2. So back to carbohydrates. So sugars like glucose, galactose, and fructose are monosaccharides. These are single unit sugars because they contain one unit of particular sugar. Okay. Glucose, galactose, and fructose are examples of simple sugars. We call them monosaccharides or simple sugars. Disaccharides is when you combine two monosaccharides with a glycosidic bond by removing water, dehydration synthesis, to make a double sugar or a two-part sugar. We call them disaccharides. Di means two. The saccharide, of course, pertains to the sugar. Important for you to remember that the component of a disaccharide or a polysaccharide for that matter, the monomer is glucose, galactose, or fructose. We'll be talking mainly about glucose, and they're joined together by glycosidic bonds. So here we go, monosaccharides versus disaccharides. So here we have glucose, galactose, and fructose, and they're all ring structures. And you'll notice that glucose and galactose are very similar. And fructose, so these are 6-ring hexagonal sugars, hexose sugars, and fructose is a pentose sugar. So if you take two glucose molecules, so if you take a glucose molecule and a fructose molecule and you link them together with a glycosidic bond by removing water, dehydration synthesis, you make the disaccharide sucrose, and you know sucrose as table sugar. So that's glucose and fructose. If you take a molecule of glucose and a molecule of galactose, you make the sugar, the disaccharide, lactose. You join them together with a glycosidic bond via dehydration synthesis, and you make the disaccharide lactose. And you know this better as milk sugar. And if you take a glucose and you link it together with another glucose molecule via glycosidic bond, you make that by dehydration synthesis, then you have the disaccharide maltose. Okay, so it would be to your advantage to remember the formulas, not the chemical formulas, but in other words, how to make the disaccharide maltose. That's glucose, lactose, that's galactose, glucose, and sucrose, that's glucose, fructose. All right, monosaccharides may occur as linear chains or as rings, and we will see them when we get to cellular respiration, for instance, as rings, but that ring has to be broken open in order to cut the sugar down into manageable pieces by the cellular respiration mechanisms. Okay, so in aqueous solutions, monosaccharides form a ring structure, and so here you see the chain structure. 1, 2, 3, 4, 5, 6. There's your 6 carbons. because remember it's a hexose sugar, and there's your ring structure, where each corner is a carbon minus this one because there's an oxygen, and your sixth carbon is hanging out there. So other carbohydrate stuff. So molecules with a 1-to-1 ratio, that is 1 carbon to 2 hydrogens to 1 oxygen ratio. These are carbohydrates, okay? So you're always going to see that standard formula, 1-to-1, carbon, hydrogen, oxygen. That is 1 carbon to every 2 hydrogens to every 1 oxygen. And so the empirical formula here is CH2O, see, that's the 1-to-1, times the subscript N is however many units of this monosaccharide you're going to be dealing with. So carbohydrates are excellent energy storage molecules. They are used mainly for energy. And there are a lot of carbon-hydrogen covalent bonds, right? And those are the bonds that are broken. And when you break a bond, a chemical bond, you release energy, as we'll see when we talk about ATP. Plants use starch for energy. Animals use glycogen for energy. Remember, I told you the first part, glycogen is the storage form of sugar, storage form of carbohydrates in vertebrate animals. Starch is the storage form for plants. Plants also use cellulose for structural support, and animals like arthropods, that's your crabs and your lobsters and your shrimp, use chitin for structural support. All of those are examples of carbohydrates. Isomers are molecules with the same chemical formula but with a different arrangement of atoms. Okay, they may be structural isomers or stereoisomers. So we're going to see different same chemical formula but different arrangement of atoms in a different order. So structural, and they're called isomers. So structural isomers, I'll show you a picture in a minute, have different arrangements of atoms. Stereoisomers are mirror images of one another. So it's like you take a molecule and you shine it in a mirror, right? It's the same, but everything is reversed. So here's a couple of examples of isomers. So fructose and glucose are structural isomers, right? Same chemical formula, but a different arrangement of atoms. And you can see where this carboxyl group of glucose, this carboxyl group up here, carbon number 1, is on carbon number 2 in fructose. Okay, so same number of atoms, same types of atoms, just a different arrangement. Now in the case of glucose and galactose, same structural arrangement, but these are stereoisomers. Okay, so they're mirror images of one another. So you could take this galactose, flip it over like a pancake, and it will lay directly on top of this glucose as a mirror image. All right, and you can see that, and so you can see how, and if you had a mirror down through the middle, this OH and that OH, all right, line up. Again, if you could flip that galactose over, it would lay right on top of the glucose, no problem. Stereoisomers. So glucose, galactose, stereoisomers, glucose, fructose, structural isomers. Okay, so back to proteins. Now, protein structure determines its function. We're going to look at several different levels of structure for proteins. Remember, protein initially is just a big long chain of amino acids joined together by peptide bonds. How do you make those peptide bonds? Dehydration synthesis, remove water. By the way, I have a handout that I made up for another class that I'm going to post for you guys as well, that talks all about it. It just reviews hydrolysis and dehydration synthesis. I'll post that tomorrow sometime. So the primary structure of a protein we see up here. Primary structure is the first level. It's just the big long chain of amino acids joined together by peptide bonds. Now, when we go into secondary structure, then we start manipulating that big long chain. And so here we see this is called an alpha helix, and we'll see this again when we talk about DNA. You may be familiar with the alpha helix already, and it's just like a corkscrew or a spring. Okay, that's secondary structure. That's called the alpha helix, and that's due to bondings between various elements of those amino acids, okay, and it causes it to corkscrew. That's secondary structure. This is called a beta pleated sheet, and I'm not concerned that you remember the details of the beta pleated sheet, but you can see how it takes on this sheet structure, and it's like sort of like slats hooked together end on end. The alpha helix, you're going to want to kind of tuck that image in the back of your mind, because we'll be talking about that when we talk about nucleic acids. But there's two secondary structures for proteins. That's the beta pleated sheet and the alpha helix. Now, when you go to tertiary structure, okay, here's the big long chain. It could be thousands and thousands, tens of thousands of amino acids, and that's the big long chain. That's primary structure. And then you start folding it on itself, twisting to make an alpha helix, or folding to make the beta pleated sheet. And then you take that secondary structure, and by the way, it could be, you could have in one part of the amino acid chain, you could have an alpha helix, and another part you could have a beta pleated sheet, as we see down here. And when you get that all together, then it becomes globular, and we call that tertiary structure. The whole bottom line here is that structure determines function, okay? So now we have this globular protein, which is ultimately a big long chain of amino acids, but it's folded on itself, and in some places you have an alpha helix, some places you have a beta pleated sheet, but it becomes a globular system. That's tertiary structure. Now, if you take a bunch of these globular proteins and you hook them together, then we have quaternary structure. So tertiary structure would be along the lines of, for instance, an enzyme. But if you take a bunch of those tertiary structured proteins and you hook them together and they become functional together, then that's quaternary structure. And so here you see 1, 2, 3, 4 of these tertiary globular proteins all hooked together. That's quaternary structure, or the fourth level of structure. And a great example, and the one that I love to use, is hemoglobin, and we'll see that in a little bit. So when a protein is unfolded such that it loses function, we say the protein has been denatured. So here we see the properly folded protein, and we denature it through some means, and then it returns to primary structure, just a big long chain of amino acids, which then, by the way, it is non-functional. It's denatured. It loses its nature in function, okay, or loses its functional nature. That's denature. And you can do this many ways, as we'll see. You can throw acid onto this thing, you can dip it in alcohol, you can heat it up. Heating is a great way, a simple way to denature proteins. Okay, so you can do this a number of different ways. But when you denature a protein, okay, you return it to its primary structure, long chain of amino acids, which again is non-functional. Okay, so proteins function best at their optimal temperatures. As I said just a second ago, you can heat a protein up and denature it. By the way, and I may have mentioned this already, once you denature a protein, you can't go back. Okay, you can always go forward when you're building proteins, primary structure to secondary structure to tertiary structure to quaternary structure. But once you have a quaternary protein or a tertiary protein and you tear it apart or you denature it, you can't go back. You can't return it to, you can't go back to primary structure and then return to quaternary structure. So human enzymes work best at 37 degrees C. That's 98.6 degrees Fahrenheit. Where do you know that from? Right, that's normal body temperature, 98.6 Fahrenheit. That's 37 degrees C. And obviously, enzymes are functional proteins. Obviously, enzymes are going to work best at normal body temperature. And here's an example that you all are familiar with. Now, when you get sick and you get a fever, a high temperature, and then you start feeling run down and your muscles ache and you feel draggy, you feel like you've been hit by a truck, that's because your proteins have been denatured. Your enzymes have been denatured. Because you get a fever, your temperature goes above 98.6, things start breaking down. Your enzymes start breaking down, and enzymes are the substance by which everything works. When your enzymes don't work, you don't work. And so that's why you feel like you've been hit by a truck, because your enzymes are no longer working. And when enzymes don't work, your muscles stiffen up, your breathing gets labored, your head starts to hurt, all of those things that happen when you get sick and when you get a fever. Optimal temperature for human enzymes, 98.6 or 37 degrees C. Very important that you remember that. High temperatures cause enzymes to denature. So when you get that fever, your enzymes start to, which are proteins, they start to denature, and then when they denature, they cease to function, and that's why you feel like crap or like you've been hit by a truck. Yes. That's why you feel like crud, sorry, when you get hit by a truck. When you have the crud, you feel like crud. Okay? So here's the little smiley-faced enzyme up there at 35 to 40 degrees C. That's optimum. Denatured proteins above 60 degrees C, they don't work, and neither do you. Proteins also function best at their optimal pH. Now, pH of human blood is very close to neutral. Your body ideally wants to be neutral. Neutral on the pH scale is 7.0. But it runs a little bit alkaline, or a little bit basic, which is above 7. 7 is neutral, so here we see 7.4, 7.3, 7.2.5, 7.25, you know, somewhere in there. Okay, very, very close to neutral. That's when human enzymes work their best. Too far above or below that nominal pH, close to neutral, your enzymes denature. And so in this little diagram you see at pH 7 this enzyme is perfectly happy, but if you go below 7, that's acidic, pH 2 here, pH 5, the enzyme starts to unravel and therefore lose function, denature. And if you go too far above neutral, which is 7, pH 8 even, which is right one step over, but remember the pH scale is jumps up by tenfold each step of the way. So pH 8 and there's pH 12, your proteins, enzymes, which are proteins, denature and cease to function properly. Okay? Chaperones are other types of proteins that help fold proteins together into their functional configuration. So when you have a long chain protein, long chain of amino acids, if you want to go to a beta-pleated sheet or an alpha helix, it's the chaperones that help that folding occur. It's interesting to me that when you extract DNA, as we'll talk about it later, but DNA is microscopic. It's inside the nucleus of a cell. A cell is microscopic, so DNA is submicroscopic because it's inside the nucleus. You can't really see it until it's time for the cell to divide. But you can extract the DNA from your cells or from a cell of your favorite fruit. You'll do that experiment in lab very shortly. And when you extract that DNA, then you can physically visualize it. You can see it in a cup or see it in a test tube, and you can, pick it up with a stirring rod. You can hold it in your hand. That comes out of your very tiny submicroscopic nuclei. How do you get that big stuff that you're holding in your hand into not only the cells, which are microscopic, but into the nuclei of those cells? Chaperones. These are the proteins that do all that work of folding everything together. Okay, so this is just a little diagram showing you the precession, the work of the chaperone. Here's the protein, right? Comes into the active site of the chaperone with a little energy, that's ATP. And then you can, this is the long chain polypeptide, you can fold it up and then it releases as a functional protein. Chaperones are responsible for doing all that work. Okay, so back to lipids. Lipids include fats and oils. Fats may be saturated or unsaturated. You've heard the term saturated and unsaturated before. What do they mean? Saturated fats have their full complement of hydrogen atoms. So a saturated fat, in a saturated fat, there are no double bonds. Okay, double bonds mean you can stuff more hydrogens in there. If everything is a single bond, a single covalent bond, that means this thing holds as much hydrogen as it can hold. It is saturated with hydrogen. That's where the term comes from. By contrast, the unsaturated fat has at least one double bond. Right? At least one. One double bond, unsaturated, because you can go in and you break the double bond and throw in another hydrogen. And so you're familiar with the terms mono unsaturated. That means it has one double bond. Versus poly unsaturated, that means there are lots, 3 or 4 or 10 double bonds in that fat. Okay, poly unsaturated. Poly means many. So as double bonds are easier to break than covalent single bonds, unsaturated fats are healthier fats. Okay, that's why when we talk about eating right, you want to limit your intake of saturated fats because you can't break those down any further. Whereas if you have saturated, or rather unsaturated, monounsaturated but better polyunsaturated fats, you can always add hydrogen, you can break those double bonds, they're easy to break, and therefore you're reducing those fats better, easier for your body to handle. Okay. So here we see the structural formula for fats. And you've seen this before. Here's your little glycerol molecule. Remember, this is a triglyceride. Here's your little glycerol molecule, 3 carbons. attached to 3 long-chain fatty acids. These are saturated fatty acids, so this is a saturated fat, versus here we have an unsaturated fat, because you can see double bond there, okay? These double bonds between carbons and oxygens don't count. You have those over here. It's these double bonds here between carbons in the long chain fatty acid. That's where you get your unsaturation from. There's one there, there's one there. So this is a polyunsaturated fatty acid. Here's a monounsaturated, and 1, 2, another polyunsaturated fatty acid, okay? You'll notice that in the saturated fats, you have these nice, long, straight, parallel chains. These little red balls right here represent the carbons in glycerol. In your unsaturated fatty acids, the double bonds cause these long straight chains to kink. So everywhere you see a little kink there, that's the presence of a double bond. Alright, and so there you see a bend. I call it a kink. A bend or a kink, that's a double bond there. And here you see boom, boom, it kinks twice, that's two double bonds. Phospholipids, a very important category of lipids that we'll be talking about for a long time. Phospholipids are major components of biological membranes. Phospholipid, by definition, it's a lipid connected to a phosphorus group, or a phosphorus atom, or a phosphate group. And remember from before, phosphate group is the P connected to 4 oxygens. So a phospholipid consists of 3 carbon, that's your glycerol molecule, with a phosphate and 2 fatty acid chains. So remember, with your glycerol molecule, you have 3 carbons, your little chain of 3 carbons, and each carbon can have 4 bonds. And so on one carbon you have a fatty acid chain, on another carbon, the second carbon, you have a fatty acid chain, and on the third carbon you have that phosphate group, the PO4. As it happens, that phosphate group, we call it the phosphate head, is polar. So phospholipids have a polarity in that the phosphate head is polar, which means that it interacts well with water. And we call those kinds of molecules hydrophilic. Hydrophilic literally means water-loving. Philo or philo comes from, I think it's Greek, that means love or to love. So hydro is the water part. So hydrophilic, water-loving. In other words, it interacts well with water. It doesn't have a problem with water. That's the polar part of the phospholipid, which is the phosphate end. Versus the lipid tail, right? These are the 2 fatty acid chains, which are nonpolar, and therefore they repel water, because you know that fats and oils do not mix with water. We mentioned that before, but you know this from your salad dressing, right? Vinegar and oil don't mix. You got to shake the bottle up before you pour it on your salad. That's because lipids and water do not mix. Lipids are hydrophobic, literally translated, that means water-fearing. And in organic systems, biological systems, that simply means that this molecule, whatever it is, does not interact well with water, does not mix with water. And so here's your triglyceride. These little junctions represent the 3 carbons of your glycerol molecule and your 3 long-chain fatty acids. By the way, these fatty acids, they can have 10 carbons in them, they can have 100 carbons in them. We call them long-chain fatty acids because they can be very long. That's your triglyceride. And in the case of a phospholipid, there's your 1, 2, 3 little carbons from glycerol. We exchange one of those long-chain fatty acids, we exchange it for a phosphate group. That's represented here in this green oval. That's your PO4. Alright, so a phospholipid is nothing but a triglyceride with one fatty acid chain removed, replaced with a phosphate group. Phospholipid. And so here we see in all of its glory in many different ways the phospholipid. So there's your little glycerol molecule, your 2 long-chain fatty acids, and your phosphate group right there. Phosphate interacts well with water. It's hydrophilic. We call that the hydrophilic head or the polar head or the phosphate head versus the lipid chains or the lipid tails, which are hydrophobic. Here's another way to look at it. There's your 3 little carbons from glycerol, your 2 long-chain fatty acids, and there's your PO4 representing your little phosphate group. We look at it in this model. There are your 3 carbons from glycerol. There's 1 fatty acid chain. This is obviously saturated, and here's another fatty acid chain with a little kink in it. That means it's unsaturated, and there's your little phosphate group up there. This is the polar end, this is the nonpolar end, this is the hydrophilic end, this is the hydrophobic end. That's really important as we talk about the cell membrane coming up. And here's a simpler, so all of these little, the dark little circles represent carbons, the white little circles represent hydrogens. Here's another simpler way to look at it. This represents the hydrophilic end, the phosphate head end, and these represent the hydrophobic or lipid tails. Okay, and remember with a phospholipid, you only have two. You have 2 tails, 2 hydrophilic, 2 lipid tails, versus a triglyceride, which has 3 lipid tails. Now, if you take a bunch of these phospholipids, just as a little experiment, you take a bunch of these phospholipids and you throw them into a beaker of water, they are going to spontaneously group together such that the phosphates, which love water, are facing outward, are in contact with water, and the lipid tails are tucked inside away from the water, because remember, they're hydrophobic. We call these micelles or myceles. So micelles or myceles form as lipid molecules are introduced to an aqueous solution, water in this case. They automatically orient themselves with their polar or hydrophilic or water-loving heads towards the water, and their non-polar or hydrophobic or fatty acid tails tucked inside of the little bubble away from the water. So you see the hydrophilic heads out here. These are your phosphate heads, and these are all of your little hydrophobic lipid tails tucked inside because they don't want any contact with water. They have actually done this in the laboratory, and these little myceles just spontaneously form. This idea of the lipids away from water and the polar heads, the phosphate heads towards water, this polarity of phospholipids is really important when we talk about the cell membrane. Okay, so here we have the phospholipid with its phosphate head, which is hydrophilic. It's attracted to water. It loves water. That's hydrophilic. And the fatty acid tails down here, which are hydrophobic. They are repelled by water. They don't like water, so they stay away from water. And so if you take a bunch and you throw them in water, they form these little mycels, these little balls with the hydrophobic tails tucked inside. When it comes to cell membranes, is where we're going with this. In this little illustration down here, so that's water and blue, water and blue. And so what that tells you is the milieu outside of the cell is aqueous or water-based, and the cytoplasm inside of the cell is also aqueous or water-based. And so what happens as a cell membrane forms, you get two layers of phospholipids, and they arrange themselves such that on both sides, the inside and outside, the phosphoheads, right, the hydrophilic heads are in contact with water on both sides, and the lipid tails, which are hydrophobic, are tucked inside, tucked between the two layers of hydrophilic phosphate groups. They form in this arrangement two layers of phospholipids with all the little lipid tails, hydrophobic tails, tucked inside, tucked in between the hydrophilic phosphate heads. Two layers form, and we call that a bilayer. Bi meaning two. All right, and that represents the cell membrane. We'll see it in detail coming up and also in the next unit. And so here's the phospholipid bilayer. The cell, also known as the plasma membrane, forms as two layers of phospholipid molecules arrange themselves such that the lipid tails point inward because they're hydrophobic. They don't want to be in contact with water. And the phosphate heads point outward because they're hydrophilic. They love water and they don't mind being in contact with water, towards the aqueous matrix or milieu on the outside and the aqueous cytoplasm on the inside. As a result, you get this bilayer formed. In fact, we call this the phospholipid bilayer. And so this is the phospholipid bilayer model of the cell or plasma membrane. And so here we see up close the phospholipid bilayer, phosphate heads on the outside, lipid tails on the inside, hydrophobic, hydrophilic out here. And so this sort of represents a generic cell. They're not perfectly round like this, but to give you the idea, outside of the cell is the water-based matrix or milieu. It's aqueous-based. It's not water, plain water, but it's water-based. It's a solution predominantly of water. And the cytoplasm inside of the cell is also a solution predominantly water. And so it should seem logical to you that the phosphate heads are going to point inward and outward towards the water, and the lipid tails are going to be sandwiched between those two layers. This is the phospholipid bilayer. Okay, back to nucleic acids. We'll talk more about phospholipid bilayers in the next unit when we talk about the structure of cells. So with regard to nucleic acids, DNA and RNA, as I mentioned back in the first part of this chapter, DNA is deoxyribonucleic acid, RNA is ribonucleic acid. Remember that DNA is ribonucleic acid that has been deoxygenated. We've taken an oxygen off. That's the deoxy part of DNA. So DNA is a double-stranded, look at that, alpha helix, right? Chaperone proteins do that work. This is secondary structure. So DNA has deoxyribose sugar. It's two strands. We call it a double helix, right? because one of these is an alpha helix. You put two together and you have two helices or a double helix. And remember, if you don't, I'll remind you that the bases between the two helices are cytosine, guanine, adenine, and thymine. RNA, on the other hand, is only single-stranded. We call it single-stranded RNA, or lowercase s RNA. And now DNA can also be single-stranded. We would say lowercase s, uppercase DNA, if we're talking about single-stranded DNA. Okay, so RNA, though, contains ribose sugar as part of its backbone. It consists of a single strand, and remember that it has the bases cytosine, guanine, adenine, and uracil, capital U. And in RNA, we replace uracil. In RNA, we replace the thymine in DNA with uracil. Okay. You'll see this in more detail as we go through nucleic acids. So here's DNA versus RNA. Once again, DNA is a double alpha helix. Right. RNA is a single strand. And we'll see more in detail the bases. But remember, C joins to G always in DNA, and A bonds to T, and these are hydrogen bonds, and that's what holds the double helix structure of DNA. That's what holds the double helices together, those little hydrogen bonds. But you have learned already that hydrogen bonds are very, very weak. However, we're talking about tons of these. We're talking about thousands, tens of thousands of these little hydrogen bonds that are holding these alpha helices together, because these helices can be many, many, many nucleotides long. Okay, thousands, tens of thousands of nucleotides long. And so when you talk about those, when you're thinking about that many hydrogen bonds, and by the way, as you can see from this diagram, there are two hydrogen bonds between T and A, and there are 3 hydrogen bonds between C and G. So everywhere you have a T and A, you have 2 hydrogen bonds. Everywhere you have a C and G, you have 3 hydrogen bonds. Multiply that by tens of thousands, and you can see how well that DNA is going to hold together. Not that it cannot be taken apart, and it can, and that's one of the things we have to do when the cell divides, but they're very easy to connect back together, hydrogen bonds. All right. In the case of RNA, you have just single-stranded, so you don't have C bonding to G and A bonding to T. You just have a single layer, right? A single strand of A's and C's and G's, and remember, no T's, but you have U's, uracil, in RNA. And so you might have a long strand. It might be A, G, G, C, A, U, A, G, U, C, C, A, U, like that, okay? And that'll make more sense when we get to transcription. All right, so the nucleotide structure of DNA and RNA, remember, and I've showed you this before, you have a nitrogenous base. This is, in the case of DNA, it's C or G or A or T. Okay, we see the double ring structure here. Why they call it a nitrogenous base? Look at all the nitrogens in that baby. Here is your pentose sugar. Remember that in RNA, this little attachment to the number 2 carbon is a hydroxyl group, an OH. And in DNA, this little attachment to carbon 2 is just a hydrogen because you've removed the oxygen. That's the deoxy part of DNA. Ribose sugar And off of carbon 5 there, you have your PO4, your phosphate group. Right? That's the basic structure of a nucleotide. Okay, 2 bonds in DNA. Look at there, I got a little animation going there. The 2 bonds in DNA. Well, first you have these phosphodiester bonds linking the nucleotides together. You see them in P there. And then remember, between the nitrogenous bases, you have hydrogen bonds. These phosphodiester bonds are very, very strong. These hydrogen bonds are quite weak. But in the case of DNA, you're talking about millions of them, potentially. There's A bonded to T with 2 little hydrogen bonds. Here's C bonded to G with 3 little hydrogen bonds. There's another A and T with 2 hydrogen bonds. And like I said, you multiply this over and over and over again, and you have potentially thousands, millions of little hydrogen bonds holding the 2 strands of DNA together. I end this part of Chapter 3 with this. This is one of the central themes of biology. We call it the central dogma. And so what this little illustration shows you, and this is something that we are going to go back to when we get to cell division, what this illustration shows you is that, first of all, notice all the arrows are pointing to the right here. So genetic information flows in only one direction, right? And it flows from DNA to RNA to protein. And once you go from here to there, you can't take a protein and reverse the procedure and make RNA and then take that back to DNA, okay? RNA is made from DNA by way of the process we call transcription. So we transcribe RNA, specifically it's called messenger RNA, or the message. We transcribe the message, the RNA message from DNA. This just shows you that DNA is double-stranded. It's got to come apart before you can transcribe it. RNA is single-stranded. All right, and then from RNA, that's the messenger RNA, lowercase m, uppercase RNA. And the process, when we take the message of RNA and you make a protein, you're translating that message. So you translate the message into the protein. We'll look at this in detail. Have a really fun activity for you to do, worksheet for you to do for that. And it's really kind of a cool process and very intricate, but quite fascinating. So information flows in one direction from DNA to RNA to protein. That's the central dogma. And the process of going from DNA and making RNA is called transcription. And the process of going from RNA to make protein is called translation. And by the way, back here you see replication, because when cells divide, they have to replicate or duplicate their DNA before they can divide. So DNA, taking DNA and making more DNA, that process is called replication. So it's called the central dogma of biology, right? So when you go from DNA to RNA, that process is called transcription. And when you go from RNA to protein, that process is called translation. We'll look more at that in the days to come, all right? And this idea of the central dogma is a major theme of biology, which will carry through into the next couple of units. All right, that's the end of this second part of Chapter 3, and that gives you enough information to tackle the Chapter 3 quiz coming up shortly. Thank you.